Okay, hello everyone. Hopefully you're having a day of some kind. I'm Tariq. I'm going to be covering urine concentration dilution and regulation of volume, the second half of it. So these are our objectives. We're going to be talking about sensor effect systems in regulating extracellular osmolality and volume. So first of all, osmoreceptor ADH system. So just to cover what ADH is, it's antidiuretic hormone or vasopressin. Now, if you'll notice here, you have the word antidiuretic over here. If you remember from CVP, one of the drugs that you may or may not have taken is diuretics, which we use to lower blood pressure. What it does is it makes you pee a lot. So antidiuretic hormone would make you pee less. Now, ADH is made in the hypothalamus. Specifically, it's made in the supraoptic nucleus and the paraventricular nucleus. Now, these are very, very long words, but you'll learn more about them in neuro. Basically, inside the brain, there's a thing called the third ventricle. It's kind of like a sack of fluid that's in the middle, and this nucleus sits on the side of this ventricle. As for the supraoptic nucleus, it sits next to the optic chiasm, where the nerves coming from the eyes pass through. So ADH is made in the hypothalamus inside these nuclei, and then it's stored inside the posterior pituitary gland. The pituitary gland is another gland that's inside the brain. It's towards the bottom of the brain. I could actually show you a diagram if I just pull a little bit of trickery. Okay, so here's a diagram of the brain. You can see that you have the hypothalamus here, which is around the third ventricle, where you'll have the supraoptic and paraventricular nuclei, where your ADH is made. Then it moves down to the posterior pituitary, which is down here, where it's going to be stored. Okay? Now, once you have triggering of the osmoreceptors that are in the third ventricle, you're going to have release of ADH. Or you have the baroreceptors that are inside the right atrium of the heart and the carotid sinus, which is in the carotid arteries. These will downregulate your release of ADH. And if you have a deficiency of ADH, that causes a disease called diabetes insipidus, which is a bit like diabetes mellitus, where you're still going to pee a lot, you're still going to want to drink a lot of water. The difference is that in diabetes insipidus, you don't have sugar in the urine, unlike where diabetes mellitus, you do have sugar. So the osmoreceptors in the brain, inside the hypothalamus, they can detect osmolality. All right? These will stimulate the hypothalamus to start producing ADH, which is then moved to the pituitary, like we said, and eventually it's released. Once it's triggered by the osmoreceptors. Now, ADH will go down to the kidney, and it's going to add more aquaporins, which are the water channels, into the late distal tubule and the collecting duct. And so with these more aquaporins, the collecting duct is more permeable to water. And so you can absorb more water, but at the same time, because these aquaporins only allow water to pass, sodium, urine, and anything else like chloride, hydrogen, isn't going to pass through. So you have the water that's absorbed, nothing else is absorbed. And so your urine is more concentrated, and the ECF, the fluid that's around the collecting duct, the fluid that's in the kidney, and later on the blood, once it's reabsorbed, is going to be more diluted. Now this is regulated, like we said, even more by the baroreceptors in the carotid arch and the right atrium. So here we have a quick diagram from one of the slides that I borrowed, where we have ADH binding here to its receptor. Now this is an adenylyl cyclase receptor, so it takes ATP and it converts it to cyclic AMP. This will phosphorylate a protein kinase, which goes down a long chain of phosphorylation, and basically it takes this vesicle of aquaporins and binds it to the surface. So these aquaporins will then move the water from the tubules, from the collecting duct, outside into the cell and later on into the blood, and then it will diffuse into the blood. And so this is another slide that I stole, where you have, here's the collecting duct without ADH. You'll notice that there's hardly any holes in there. And then you'll look with ADH, you'll see all these little pores where water can pass through. Now, this again as a diagram, just to recap how we synthesize and release ADH. You have the osmoreceptors in the third ventricle over here. These will cause the hypothalamus in the supraoptic nucleus to produce ADH. So here's the neuron. It's going to move down to the posterior pituitary, and then it's going to be released into the blood. Okay. Now you also have these baroreceptors over here in the right atrium and in the carotid sinus. What these are going to do is they're going to control the production of ADH in these nuclei. And so what they're going to do is if you have high pressure, if you have high blood pressure, the neurons here are going to activate. You're going to have a release of ANP from the right atrium, and you're going to have activation of the nerves from these baroreceptors. And then these are going to tell the osmoreceptors, hey, we have a lot of blood pressure, we have a lot of fluid. Even if you have high osmolality, I want you to ignore that signal. And so these will down-regulate the effect of the osmoreceptors on the release of ADH. So again, just to put it one more time, your baroreceptors will signal the osmoreceptors, will signal the nuclei to ignore the signal from the osmoreceptors, which produces less ADH. And so here's just a cleaner diagram if you want to look at it. I'm going to skip ahead. So these are the stimuli that control the release of ADH that we described before. So first you have osmolarity, where once you reach about 280 milliosmolar per kilogram of water, you're going to start seeing a release of ADH. Okay, and similarly with blood volume, once you drop below 10% of blood volume or blood pressure, you're going to see more release of ADH. But again, just to bear in mind, blood volume and blood pressure doesn't tell the brain to release ADH or stop releasing ADH. It tells the brain whether or not, or how much attention it should pay to the osmolality signals. So here's the effect of ADH on urine production. ADH, like we said before, it only controls the reabsorption and excretion of water. And so what you're going to notice is that if you have more ADH, you're going to absorb more water, and so less urine passes. What this means is that the solutes, so stuff like urine, sodium, chloride, aren't under the control of ADH at all. Now, normally, what does control them is a different hormone, like aldosterone. And here's a quick diagram that looks at the fluid intake and water loss. Now, the key thing that I want you to notice is that we intake, or we drink, as much water as we get rid of, okay? So we drink water, there's also water in food, and finally we produce water in our body through the breakdown of glucose. And then these are the ways where you lose the water, and you'll notice that the amount totals to about the same. And this is how different temperatures and different conditions will affect that intake and excretion. So you'll notice that in normal temperatures, we have the same diagram as before. But once you have hot weather, the skin, it loses the same amount of water as before. The lungs lose less water. Now, this sounds like it doesn't really make sense, but you don't really have to know the general idea. All you need to know is that hot weather lowers the amount of water lost in the lungs, but it increases the amount of water lost as sweat, okay? So as compensation, the amount of water lost in urine goes down. But the total water loss has increased, and so we need to drink more water to compensate. On the other hand, if you are like working hard, such as if you're running or if you're doing a lot of hard work, what you're going to notice is that the lungs now release less water. They lose less water through the lungs than before. And the sweat, you're going to notice that there's a lot more that's lost compared to before, right? Because again, you're working hard, you're sweating a lot, you're trying to cool yourself down. And so what this means is that the urine has to now become much more concentrated because your kidneys are trying to conserve water so that you're not losing as much water. But at the end of the day, you're still losing much more water than you were before, and so you have to drink more water to counterbalance. And so these are the two ways in which our intake of water is controlled. You have thirst and you have salt hunger. Okay? Now both of them are detected by the hypothalamus. But thirst is triggered by intracellular dehydration, right? Kind of a confusing word, but basically you have high sodium in the ECF around the osmoreceptors and around the nerves. And so this is going to pull water out of the cells, which causes the cells, the intracellular fluid, to become more dehydrated. Now the threshold for this is if the osmolarity increases by more than 4 milliosmolar. On the other hand, salt hunger is stimulated by low blood volume and by low sodium inside the extracellular fluid. And so what the low sodium and extracellular fluid does is, now that we have low sodium, so you have a high amount of water and a much higher osmolarity inside the cells. And so this is going to push water into the cells. And so you'll develop salt hunger. Now the thing is to compare these two is that thirst happens very quickly. You'll notice that if you go for a run, you get thirsty very quickly. Whereas salt hunger, you'll hardly ever notice it, if you even do, because it develops after hours and days. Now, here's what happens to blood volume when you compare it to extracellular fluid. In this first portion, you'll notice the person's just completely dead because there's not enough fluids inside the body to support life. As your extracellular fluid increases, the blood volume is going to increase to match it. Okay, because what you're going to have is you're going to have more blood volume, and so there's more water outside the cells in general, and this will also push more water into the interstitial fluid. But at some point, which is over here, your blood vessels are going to reach their maximum capacity. And so you can't put any more water inside the blood vessels. And what this is going to do is it's going to mean that all of that water has to go somewhere. And so it's going to go into the interstitial fluid. And with a lot of water going into the interstitial fluid, you're going to get edema. So here's a bunch of regulators of blood volume. We spoke about ADH before, and here's a bunch of others. You have pressure diuresis, which is the most important, and it's what we're going to talk about next. And you have all these other nice looking mechanisms. So first of all, pressure diuresis, which is a very fancy word, and it's a very weird word, but all it's really saying is that the higher your blood pressure, the more urine you're going to be producing. Okay, now how this happens, nephrologists are kind of nerding out, but they're not really getting a clear answer. But the general idea is that you're going to have more blood flow through the vasorector, which washes out the medulla, and it provides fresh extracellular fluid around the loop of Henle. And so the loop of Henle can basically pour more solutes into the extracellular fluid, which means you can lose more water afterwards. The other way is that sodium hydrogen exchanges are put into the proximal tubules. How does this happen? We're not really sure, but we know that it does happen. So here's a very, very fancy schmancy diagram that I'm not particularly a fan of. So I'm just going to pause for a second. Okay. Okay, so we have, this is the mean arterial pressure. Now it's controlled by two things: the total peripheral resistance and the cardiac output. Okay, and so the cardiac output, the total peripheral resistance is controlled by arteriolar constriction. Okay, as well as hematocrit, but that's not really as important here. Now, if your arterial pressure falls, this is going to activate the pressure receptors in the arteries, and this is going to lead to activation of the sympathetic nervous system. Okay, what the sympathetic nervous system is going to do is it's going to cause more arteriolar constriction, which increases tissue peripheral resistance and thus increases arterial pressure. Now, the other thing that happens is the activation of the sympathetic nervous system is going to cause constriction of the veins. Okay. Now this is going to increase venous pressure, which pushes more blood into the heart and increases stroke volume. Now, the final thing that it's going to do is it's going to carry a positive chronotropic effect, and that also will increase cardiac output. Okay, and so what happens here is that you're going to have, basically, you're going to have higher stroke volume and the heart is going to beat faster, which will increase cardiac output, increasing your arterial pressure. Now here we have the natriuretic peptides, ANP and PNP. Okay, these are produced in the heart inside the atrium and the ventricles. Okay, these are stimulated by guanylyl cyclase receptors. And so what happens is these will produce cGMP, which is a bit like cyclic AMP, but it's a bit fancy, it's cyclic GMP. And so these will lower tissue plasminogen resistance, total peripheral resistance, and it will increase the excretion of sodium and water. Okay? And so what these will do is if you have high blood pressure, okay, these are going to be released. These will lower these will lower total peripheral resistance, and so this will lower blood pressure, and these will increase the excretion of sodium and water, which also lower blood pressure. The other thing that these do, that these two molecules will do, is that they're going to limit the secretion and effect of ADH. Like we said before, ADH increases blood pressure, and so by limiting the effect and secretion of ADH, it's going to lower blood pressure. And then you have urodilatin, which is from the distal tubule and the collecting duct. These are stimulated by a rise in blood pressure and a rise in extracellular fluid. What these will do is that they'll cause reabsorption of water and sodium in the collecting duct. Okay, these will inhibit reabsorption of water and sodium, of water and salts in the collecting duct. Now, you might remember from before that we said that ADH only controls water. So this will inhibit the reabsorption of water and sodium as well in the collecting duct. This one has a stronger effect than ANP, and the reason for that is that ANP is degraded inside the kidneys, whereas urodilatin is not. Finally, you have uroguanylene and guanylene. What these basically do is that when you eat salt, it sends a signal from the GI tract, and know, hey, this person has eaten salt, let's get rid of salt to compensate for it. And so what it's going to do is it's going to lower salt and water reabsorption. And so this is the activity of renin. What it's going to do is it's going to reduce renal perfusion, and it's going to cause the production of angiotensin II, which is another molecule that we'll look at in a bit. So what happens is if you have low renal perfusion or you have hyponatremia, this will cause the kidneys to secrete renin, which is going to carry on the effect. And here's angiotensin II. Now, what angiotensin II is going to do is it's going to constrict both the afferent and the efferent arterioles inside the glomerulus. So here's the glomerulus. So this one will get constricted a little bit, whereas this one gets constricted a lot. Now, what this does is it lowers renal blood flow, first of all, but it also increases the filtration fraction. And what that means is that more water is going to get taken out of the kidneys. Okay, and so what it's going to do is that you're going to have later down the line is that you're going to have higher reabsorption of sodium. It's going to have higher reabsorption of sodium. Sorry, 1 second. And by extension, higher excretion of sodium and water. Lower excretion of sodium and water. The other thing that angiotensin does is that it lowers medullary blood flow in the vasorecta. And so, like we said, the vasorecta are the blood vessels that surround the loop of Henle. And because the extracellular fluid isn't getting washed as much, this is going to cause higher concentrations of solute in the medulla, which is going to lower renal sodium and water excretion. Okay, here's a quick summary of all of the hormones and what they do to the reabsorption of sodium and water. And here's another summary that tells you what each hormone does, what stimulates it, where it's produced, and its effect. I'm not going to go over them because honestly, I'd just be reading through it. And that is all. Thank you very much, and I hope you have a nice day, and best of luck on your studies.